Hi everybody, welcome or welcome back to my channel. This is Sagnick Dutta. I'm a second professional MBBS student at Burdwan Medical College and Hospital, West Bengal. In this series of videos, we'll be talking about hematology, which is basically a portion or a chapter of physiology, which is usually being taught in the first year of medical college. Now, hematology is a very important topic or a chapter, not because it is vast, but because it holds a lot of clinical importance. Some of the most common clinical cases found in India are related to blood and hematology itself, for example, jaundice and anemia. All these will be dealt in detail in these series of videos. So without much ado, let's get into the screen and start studying. So let us discuss about hematology. Now, what actually is hematology? Hematology is the study of blood. Now, blood is the fluid connective tissue in the body which helps in the transportation of different substances, especially oxygen, to different tissues and cells of the body. Now, some general considerations of the blood. Blood is made up of some formed elements, that is the cell, as well as plasma. Now, talking about the formed elements basically consist of the erythrocytes, the leukocytes, and the thrombocytes, that is basically the platelets. Right. Now, in this video, we'll be talking specifically about the erythrocytes. Now, talking about the erythrocytes are basically the red blood cells. Now, red blood cells are very typical in their characteristics. For example, these are non-nucleated. These do not have any membrane bound organelles such as mitochondria. They do not have mitochondria. Now, the absence of mitochondria is very obvious because the main function of RBC is to transportation of oxygen. Now, if mitochondria would have been present in the RBC, the oxygen carrying capacity of RBC would have decreased because the mitochondria of its own self would have used the oxygen present in the RBC. Now, other than these characteristics, the RBCs contain hemoglobin, and apart from these, they also have got a shape of biconcavity. They have got a concave shape. Right, these are the basic characteristics of RBC. Now, the general basic lifespan of RBC is around 120 days. Right. Now, the main focus area while studying erythrocytes is the formation of RBCs, which is also known as erythropoiesis. Now, erythropoiesis is basically the process by which RBCs are formed in the body. Now, talking about the formation of RBC or erythropoiesis, it occurs in different organs at different point of life, starting from the developing phase to the adult phase. For example, in the first trimester, the erythropoiesis occurs in the mesoderm of the yolk sac and thus is known as the mesoblastic stage. Now, in the second trimester, it occurs in the fetal liver and spleen and thus is known as the hepatic stage. And talking about the third trimester, it occurs in the fetal bone marrow. Right, so fetal bone marrow, so this is known as the myeloid stage. Right. But at birth or after birth, all the bones have the child have got the red bone marrow. But soon after, when the child grows and goes towards becoming an adult, what happens is only few bones have got the red bone marrow because this red bone marrow is being converted into an yellow bone marrow. As a result, the sites of production of red blood cells decreases. So most of the red blood cells in adult life are being formed at the proximal ends of long bones. They also occur in the sternum and some of the flat bones, vertebral bodies, as well as some anterior superior iliac spine. Fine. Now the most important thing that we need to know is the steps of erythropoiesis. Now the steps of erythropoiesis starts from the developmental stage itself. The first thing that is formed is the pluripotent hematopoietic stem cells. The pluripotent hematopoietic stem cells are then differentiated to form some committed stem cells. One of the committed stem cells forms the erythroid series, and another committed stem cells group forms the lymphoid series. Right. Now these lymphoid series, what happens is these go out of the bone marrow, and these erythroid series remains in the bone marrow and thus helps in the formation of different blood cells. These erythroid series of committed stem cells, they form three different types of colony forming units. The first one being the colony forming unit of erythrocytes. The second one is the colony forming units of megakaryocytes, and the third one is the colony forming unit of the granulocyte and monocytes. The colony forming units of erythrocytes, they differentiate to form the proerythroblast, which in turn differentiates to form the early normoblast. The early normoblast differentiates to form the intermediate normoblast. The intermediate normoblast then differentiates to form the late normoblast. After all these normoblastic stages, reticulocytes are formed, which finally differentiates to form the final matured erythrocyte. Right. Now, some basic information or a very prime information which can be asked is, at which stage of this erythrocytic development does blood hemoglobin starts to appear? The answer shall be the intermediate normoblast. In the intermediate normoblast, hemoglobin begins to appear in blood. Now, let us just get to know some general characteristics of all these stages. Now, the first stage or the first characteristic that we need to know is about the cell size. If we talk about the very first stage, that is this PHSC, it has got a diameter of around 24 microns, whereas a mature erythrocyte has got a diameter of around 7 to 8 microns. So what basically can be seen is that cell size goes on decreasing while moving from the most immature cell to the most mature cell. And there's a specific reason to that. What is the reason? For example, this is a immature RBC. This differentiates to form two daughter cells, and these each cell differentiates to form two daughter cells each. Now what actually happens is, while these daughter cells are not being totally matured, they start dividing to form the other two daughter cells. So what happens basically is cell division and cell maturation are occurring simultaneously in the bone marrow. As a result, the daughter cells have very little time to grow as much as the parent cell. As a result, what happens is the size goes on decreasing from the PHSC being a 24 micron diameter to the final erythrocyte being around 7 to 8 microns. Talking about the second property, that is about the nucleus. Now, if we talk about the immature RBCs around the PHSCs, the normoblast, they have got some nucleus. But a mature RBC do not have the nucleus. The last nucleus can be seen in the late normoblast state. But this nucleus is characteristically very pycnotic or eccentric. What is meant by pycnotic or eccentric is that these are peripheral nuclei, like a very small invisible nuclei present at the periphery of the cell. These are known as pycnotic or eccentric nuclei. Right. Now talking about the third characteristic is the cytoplasm. Talking about the cytoplasm, in the earlier stages, the cytoplasm is basophilic. That is, the contents are acidic and they attract the basophilic stain. But finally, it becomes acidic or acidophilic. That is, the contents become basic. Now, when we have talked about the salient features of different stages of erythrocytes, let us talk about the factors determining the rate of erythropoiesis. The very first factor and the most important factor of all is tissue oxygenation, or we may phrase it as hypoxia. Like hypoxic condition actually regulates the rate of erythropoiesis greatly. This is the most important regulator of RBC production. Now, what is hypoxia? Hypoxia is any condition when the oxygen level decreases to the tissues. The oxygen level which is being transported to the tissues decreases. For example, at high altitudes where the air pressure is low, the oxygen pressure is low, what happens is less oxygen is available for the body. So the rate of erythropoiesis increases to compensate for the less amount of oxygen, so that more and more RBCs are produced, so that more and more of oxygen is being transported. Second case, in case of any heart or lung disease, serious diseases of these two organs result in deficient oxygen supply. To cope up with all these, what the body does is it increases the rate of erythropoiesis. What occurs in all these cases is the hormone erythropoietin is being stimulated, which increases the rate of erythropoiesis. The second important factor controlling the rate of erythropoiesis is the hormone erythropoietin itself. The erythropoietin hormone is being synthesized by the kidney mostly. That is, 90% of it is being synthesized in the kidney, whereas in the liver 10% of it is being synthesized. Now, when there are hypoxic conditions, the interstitial cells of the kidney, they secrete erythropoietin into the blood. Now, these erythropoietin reaches the bone marrow. At the bone marrow, there are some specific receptors, that is EP1 and EP2, on which the erythropoietin acts. It stimulates the rate of erythropoiesis. Specifically, the conversion of the hematopoietic stem cells, the PHSCs, to the pro-erythroblast occurs faster. The other steps are also increased, this rate of the other steps are also increased as well, but the conversion of the PHSCs to the pro-erythroblast is highly increased. As a result, the production of RBCs increases. For example, the erythropoiesis process takes around 7 to 9 days, but in this case, due to the increase in the erythropoietin secretion, it is decreased to around 5 to 6 days. Right. Now the next important factor are some dietary factors. Now understand this properly. RBCs contain hemoglobin. Now this hemoglobin is made up of heme and globin. Heme contains iron and globin contains some amino acids. Right. So a diet deficient in iron would result in decreased hemoglobin. A diet deficient in some essential amino acids required for the production of globin would result in the decrease of hemoglobin. So these are some common things. Now, vitamin B12 as well as folic acid, which is basically vitamin B9, are also very much required in the diet for some sufficient production of RBCs. Look, RBC is a non-nucleated cell, but its precursors, the PHSCs, the erythroblasts, all are nucleated cells. And vitamin B12 and folic acids are required for the synthesis of DNA. If these are absent, DNA secretion is hampered. As a result, nuclear maturation in the RBC precursor fails. So the cells will remain large and immature, resulting in megaloblastic anemia. We will discuss about megaloblastic anemia separately in the anemia video, but this is the basic concept of the formation or occurrence of megaloblastic anemia. Now there are some other factors such as vitamin B1, B2, B6, some minerals like copper, cobalt, zinc. These also controls the formation of RBCs, that is the process of erythropoiesis. Fourth factor are the hormonal factors. The first thing that we need to talk about is the testosterone, which is prevalent in males. As a result, erythropoiesis is increased in males. So the volume of RBC is more in males. In male, the sex hormone stimulates erythropoiesis by stimulating the erythropoietin production from the kidneys. Secondly, it potentiates the action of erythropoietin to act on the bone marrow, and it directly also stimulates the process of erythropoiesis. And in females, estrogen is considered to decrease the process of erythropoiesis. This we come to the end of the factors regulating the rate of erythropoiesis. Now let us do some clinical significance of erythropoiesis. There are some drugs, for example, chloramphenicol or some radiations. These decrease the rate of erythropoiesis. Some persons residing at high altitudes, they have increased RBC's volume, and the reason is being explained here due to hypoxic conditions. Here we have explained that due to hypoxic conditions, in air pressure is low, oxygen pressure is less. As a result, what happens is RBC production is increased. Now, erythropoietin which is a hormonal factor controlling the rate of erythropoiesis, is synthesized in the kidneys. Now consider a patient having a chronic renal failure. They will lack erythropoietin. As a result, what will happen is the RBC production will decrease. Right? RBCs do not have some membrane bound organelles like mitochondria and ribosomes. Now, there must be a question that how does then RBC perform its metabolic activities? What happens is RBCs have got some cytoplasmic enzymes which actually help in the metabolization of glucose and produce ATP. The enzymes help in maintaining the flexibility of the red cell membrane and keep the iron in the Fe2 plus, that is ferrous state. The ATP is required in maintaining the function of the sodium potassium pump. The sodium potassium pump is being maintained by the ATP formed in the red cell membrane. Now, while towards the end of the lifespan of these red cell membranes, what happens is these enzymatic system, these start to get exhausted. As a result, the red cell membrane becomes more fragile. Now, the cell gets easily ruptured while passing through some capillaries. As a result, these are destroyed in the spleen. The splenic macrophages swallow these fragmented RBCs. As a result, hemoglobin is liberated. So this brings us to the next topic of discussion, the metabolism of hemoglobin. When hemoglobin is released by the breakdown of RBCs, these are broken down into heme and globin. The heme is converted to biliverdin and Fe2 plus is released. This Fe2 plus gets into the iron pool of the body, and globin is converted into amino acids and is thus sent to the protein pool of the body. Now this biliverdin is converted to bilirubin. In the peripheral blood, bilirubin attaches with albumin and forms a bilirubin-albumin complex. This is sent to the liver. In the liver, conjugated bilirubin is formed, which is then sent to the second part of duodenum, where some part is being converted to stercobilinogen, which is then oxidized to form stercobilin and are excreted in the stools. This actually gives the yellow color of the stools. Now some of it is also excreted as urobilinogen through the urine. So this is all about the metabolism of hemoglobin. Now let us discuss a clinical condition related to this, which is known as jaundice. Jaundice is something very common metabolic condition which is found in your clinics. Now how do we actually define jaundice? Jaundice is the yellowish discoloration of the mucous membrane and skin, which is caused due to the increase in bilirubin levels of the blood. Jaundice becomes clinically apparent when this bilirubin concentration exceeds 3 milligram percentage. Now the normal bilirubin percentage is around 0.3 to 1 mg percent. So if this increases 3 mg percent, what happens is termed as jaundice. Jaundice is detected clinically by the examination of the sclera. Now, why is this sclera preferred? Because it can be easily made out when a white sclera, the sclera is usually white, right? So that turns yellow is usually seen and very effectively identified. And moreover, the sclera contains a protein called as the elastin, for which bilirubin has got a high affinity. And that is why sclera continues to be in a yellow color for a long period of time even after jaundice has been cured. Now, depending on the cause, the jaundice is said to be of three types. The first one is the pre-hepatic jaundice. In case of pre-hepatic jaundice, the abnormality lies before the bilirubin goes to the liver. There is the problem before it goes or enters into the liver. There is some excess breakdown of RBCs. As a result, excess liberation of hemoglobin and excessive formation of bilirubin from the heme. So the bilirubin increases in the blood, hence is also known as the hemolytic jaundice. Second type of jaundice is known as the hepatic jaundice. The abnormality lies in the processing of bilirubin once it has reached the liver. That is, there is a problem in the conjugation of the bilirubin. For example, in case of disease of liver parenchyma or any injury to the liver cells will cause bilirubin to enter blood and directly from the liver. For example, in cases of hepatitis A, B, and other kinds of cirrhosis. Now, the third type of jaundice is the post-hepatic jaundice. What happens in post-hepatic jaundice is, for example, once the bilirubin is conjugated in the liver, it is excreted in the bile. Then via the bile ducts, it reaches the intestine. Now, if there is obstruction in the bile ducts, the bile ducts are obstructed, what happens is the bile and the bilirubin will not reach the intestine. Usually, this reaches the intestine through the bile ducts, but that won't happen. As a result, the bilirubin will start entering directly into the blood. As a result, this is also called as obstructive jaundice. Right. Now, since bilirubin is already conjugated by the liver, it is the conjugated bilirubin that increases in the serum. The stools become chalky and pasty. The conjugated bilirubin is measured in this case, and a confirmed case of post-hepatic jaundice is said to be present. Now, let us discuss a little details about hemoglobin. Now, what actually is hemoglobin? Hemoglobin is a conjugated chromoprotein, this is a conjugated chromoprotein, which is present inside the RBCs. It contains iron in the form of Fe2+. It is due to hemoglobin that the red blood cells and the blood are red in color. Its main function is to transport the oxygen to the tissues. Important role in acid-base buffering in the extracellular fluid is also performed by the hemoglobin. Talking about the normal level of hemoglobin, it is around 14 to 18 gram percent in male and around 12 to 16 gram percent in females. The oxygen carrying capacity of blood or hemoglobin is that 1 gram of hemoglobin carries around 1.34 milliliter of oxygen. Hemoglobin is made up of heme and globin. Heme consists of iron. There are 4 globin chains, 2 alpha chain and 2 beta chains in adult hemoglobin. Now, if we talk about the synthesis of hemoglobin, which is basically dealt in biochemistry, but we will talk about it in short. What happens is 2 molecules of succinyl CoA combines with 2 molecules of glycine to form a pyrrole ring. 4 molecules of pyrroles forms protoporphyrin 9. A molecule of protoporphyrin 9 combines with Fe2 plus to form the heme moiety. Now the heme plus the polypeptide forms the alpha or beta hemoglobin chain, and 2 alpha plus 2 beta forms the adult hemoglobin. Now there is a specific kind of hemoglobin which is also known as the glycosylated hemoglobin, which is also represented as HbA1c. This is also known as the glycated hemoglobin. What happens is glucose in the plasma gets attached to the hemoglobin non-enzymatically but irreversibly. What happens is glucose plus hemoglobin gets attached and forms HbA1c. The percentage of hemoglobin that is in the glycated is a very good indicator of the plasma glucose over a certain period. If plasma glucose is consistently increasing, more of glucose will attach with the hemoglobin. If plasma glucose is increasing, more of it will attach with the hemoglobin, and as a result, more of glycated hemoglobin will form. A percentage of around 6 of glycated hemoglobin is normal. Once the glucose is attached to hemoglobin, the hemoglobin remains glycosylated for the remainder of the lifespan of the red cell. Hence, glycosylated hemoglobin indicates the average plasma glucose level over a 3-month period, that is 120 days, which is the average lifespan of the RBC. So, blood glucose sugar level can be measured by the presence of glycated hemoglobin. I hope this video has been very clear and concise for you all. The important topics have been dealt in here related to the erythrocytes. In the upcoming videos, we will discuss about the leukocytes, the blood indices, anemia specifically, which is the most important part of the hematology section, and many other clinical correlations as well. So if you really like the video, hit the like button, subscribe to the channel, and share this video with all your friends and family, whoever find it helpful. Thank you.